Imagine for a moment that you are watching the greatest rock band in history. For decades, the spotlight has been entirely on the lead singer, charismatic, famous, the one everyone talks about. Let's call him DNA. He's the blueprint, the star, the one we think runs the show. Standing in the shadows, barely lit, is the backup singer, quiet, unassuming, just there to fetch water and hand over the microphone. We call her RNA. For nearly half a century of modern biology, that is exactly how we saw the world. DNA was the boss, the master architect, and RNA was just a lowly messenger, a fetch quest lackey. But what if we were wrong? What if the backup singer was actually writing all the songs, playing all the instruments, and yet this was actually the one who started the band in the first place? Welcome to the show. Today we are diving into the catalyst, RNA, and the quest to unlock life's deepest secrets by Nobel laureate Thomas Cech. And Alex, get ready to have your mind blown because we are entering the age of RNA.
Okay, Sarah, you know I love a good underdog story, but that is a massive claim. I mean, I learned in high school biology, and I think most of our listeners did too, that DNA is the king. It holds the genetic code. It's the blueprint of life. You're telling me that RNA, the thing that just copies DNA, is actually more important? I'm skeptical. I mean, sure, we've heard about mRNA vaccines recently, so obviously it does something, but the origin of the band? That sounds like a stretch. But I am intrigued by this book because Thomas Cech isn't just some science writer, he's a Nobel Prize winner who literally changed the textbooks. So, if we are in the age of RNA, as he puts it, what does that actually mean? Are we saying physics is out and biology is in? Or is this just a rebranding exercise for a molecule that's been jealous of the double helix for too long? Break it down for me.
It's definitely not a rebrand. It's a revolution. To understand why, we have to go back to the 1,950 seconds. DNA had just been discovered, the double helix, Watson and Crick, the whole deal. It was huge. But there was a problem. They had the code the A, T, C, and Gs, but they had no idea how that code actually turned into a living, breathing organism. It's like finding a book written in a language you can't read. You know it's important, but you can't build a house with it. Enter George Gamow. He was this towering, eccentric physicist, a practical joker who loved magic tricks. He formed this exclusive group called the RNA Tie Club. I'm serious. They literally wore wool ties with RNA sequences embroidered on them. They were trying to crack the code using math. They knew DNA lived in the nucleus, but proteins, the stuff that actually builds your body, were made outside the nucleus. So, they hypothesized there had to be a messenger, a go-between. That was the first hint that RNA wasn't just a copy. It was the essential link.
Wait, wool ties? That is fantastic. But okay, so they have a theory. DNA is in the castle tower, the workers, are in the field, the cytoplasm, and someone needs to run orders between them. That's the messenger RNA, or mRNA. But finding it wasn't easy, right? The book talks about this incredible experiment by Sidney Brenner, Francois Jacob, and Matt Messelson. It sounds like a spy novel operation. They were looking for this phantom molecule. The problem was, when they looked at cells, most of the RNA seemed stable and permanent, like the furniture in a factory. But if you change the orders, say, tell the bacteria to digest a different food, the production changes instantly. So the messenger had to be fast, temporary, and volatile. They finally found it by infecting bacteria with a virus and catching the fleeting moment when the virus injected its own orders. It was the aha moment. The ribosome is just a record player. It'll play whatever record, whatever mRNA you put on it. That's a powerful analogy.
Exactly. The ribosome is the turntable, and the mRNA is the vinyl record. You can swap the record, and the music changes. That discovery was monumental, but they still couldn't read the language. They knew the letters A, U, C, G, but they didn't know the words. This brings us to Marshall Nierenberg. He wasn't part of the elite Tie Club. He was an outsider, working on his own. He did something brute force and brilliant. He made a synthetic RNA made of times only times uracil poly-U. Just UU forever. He fed it to the ribosomes, and out popped a protein made of only one thing: phenylalanine. Boom! He cracked the first word. UUU means phenylalanine. It was the Rosetta Stone of biology. Suddenly, the code wasn't a mystery. It was translatable. This era was all about defining RNA as information. It was the messenger era. But honestly, Alex, that's the boring part compared to what comes next. Because up until the late 70s, everyone thought RNA was just a passive script. It didn't times do times anything.
Right. And this is where the story gets really weird. Splicing. I was reading this part and thinking, why is nature so inefficient? So, apparently, in human cells, the DNA gene isn't just one long continuous sentence. It's interrupted. It's like reading a magazine article where every other paragraph is just gibberish that you have to cut out with scissors to understand the story. These interruptions are called introns. When the DNA is copied into RNA, the introns are still there. Something has to come along and physically cut them out and stitch the good parts, the exons, together. This was the bombshell at Cold Spring Harbor in 1977. But here's my question, Sarah. If you have to cut and paste the RNA every single time, you need a pair of scissors and a bottle of glue. In biology, scissors and glue are enzymes. And everybody knows or knew that enzymes are proteins. So they started looking for the protein that did the cutting. That's just logic, right?
It was logic. It was the central dogma of biochemistry. DNA stores info, RNA carries info, proteins do the work. Enzymes are proteins. Period. End of discussion. If you have a chemical reaction happening like cutting RNA, there must be a protein enzyme doing it. This was the world Thomas Cech lived in when he started his lab in Colorado. He wasn't even trying to revolutionize biology. He was just studying this weird little single-celled pond creature called Times tetrahymena asterisk. He wanted to understand how its ribosomal RNA was made. He hired a technician with golden hands, Art Zaug, and they started purifying the RNA. They knew there was an intron, a piece of junk code that got cut out. So they set up the experiment to find the protein scissors. They isolated the RNA, they added nuclear extract, which should contain the protein enzymes, and boom, the intron was cut out. Success! Science works. But then they did the control experiment.
The control experiment. This is the part that gave me chills. For those listening, a control is where you leave out the key ingredient to prove that nothing happens without it. Like trying to bake a cake without flour to prove flour is necessary. So, Cech and Zaug take the RNA. But this time, they leave out the nuclear extract. They leave out the proteins. They expect nothing to happen. The RNA should just sit there, unspliced. But when they looked at the results, the splicing still happened. The intron was gone. The RNA had cut itself. I can imagine Cech staring at that X-ray film. It's impossible. It's like leaving a pile of wood and nails in a room, coming back an hour later, and finding a chair built with no carpenter in sight. They must have thought they messed up, right? Contamination? Maybe a tiny bit of protein was stuck to the RNA?
Oh, they absolutely thought they messed up. Cech describes it like plucking petals off a daisy. It's a protein. It's not. It's a protein. It's not. He spent a whole year trying to find the ghost protein. They boiled the sample. Proteins die when you boil them. RNA doesn't. The reaction still worked. They used detergents that destroy proteins. Still worked. Finally, they did the ultimate test. They engineered the RNA in a bacterium that had never seen times tetrahymena times before, so there could be no times tetrahymena times proteins present. Pure, synthetic RNA. And it spliced itself. That was the moment the world changed. RNA wasn't just a passive scroll of text. It was a pair of scissors. It was an enzyme. Cech coined the term ribozyme ribonucleic acid enzyme. It sounds technical, but Alex, this shattered the protein monopoly. It's like finding out the book can read itself.
And he wasn't alone, right? Around the same time, Sidney Altman at Yale was studying this other enzyme called RNase P. It cuts tRNA. Everyone told his grad student, "You're bad at purifying this enzyme because there's always this piece of RNA stuck to it." They thought the RNA was a contaminant. But Altman realized the RNA wasn't trash. It was the engine. The protein was just the frame. This earned them both the Nobel Prize. But here is where my brain starts to hurt a little. If RNA can fold up and act like a machine, it must have a shape. Proteins are like complex origami. They have pockets and arms to grab things. I always pictured RNA as a wet noodle, just a floppy string. How does a wet noodle act like a pair of scissors? Form has to follow function, as the architects say. So, what does this ribozyme actually look like?
That is the perfect question. You are right. Usually, we think of RNA as a linear string of letters, but RNA is sticky. An A loves to stick to a U, and a G loves a C. So, the string folds back on itself. It makes loops, hairpins, and knots. It becomes three-dimensional. Getting a picture of this was incredibly hard. It took decades of work, specifically X-ray crystallography, which is like trying to figure out the shape of a chandelier by shooting a laser at it and looking at the shadow. Jennifer Doudna, who later became famous for CRISPR, actually started her career in Cech's lab trying to crystallize this times tetrahymena, times ribozyme. When they finally saw the structure, it was beautiful. It looked like a protein. It had a core, it had crevices, it had a specific shape that could grab another molecule and snap it. It wasn't a noodle. It was a machine made of code. And this realization led to the biggest shocker of them all, the mothership.
The mothership, the ribosome. This is the factory that makes all times the proteins in your body. It is massive. And for the longest time, scientists thought it was a protein machine that used a little bit of RNA as scaffolding, like steel beams holding up a brick building. But Harry Noller, this jazz-playing, Ferrari-fixing scientist, thought differently. He suspected the RNA was the main actor. He stripped away the proteins, and the ribosome still worked, mostly, but proving it required seeing it. The race to get the structure of the ribosome is epic. We're talking about the Queen Mary of molecules here, huge, complex. When Tom Stites and his team finally got the high-resolution image in 2000, they looked at the active site, the place where the amino acids are actually stitched together to make life. And guess what they found? No proteins, none, zero. It was pure RNA.
This is the mic drop moment of biology. The machine that makes proteins is itself an RNA machine. It's a ribozyme. This means that at the very heart of life, before there were cells, before there were animals, there was RNA. This solves the biggest paradox in science, the chicken and egg problem. Here is the paradox. DNA holds the instructions to make proteins, but you need protein enzymes to read DNA and replicate it. So, DNA needs proteins, and proteins need DNA. Who came first? It seemed impossible. But now we know RNA can do times both asterisk. It can store information like DNA, it has a code, and it can cut, splice, and build like a protein. It's an enzyme. So, the answer to chicken or egg is neither. It was the RNA world. In the primordial soup, 4 billion years ago, RNA was the Swiss Army knife that started it all. It bootstrapped life.
That is wild. We are essentially walking, talking bags of RNA that evolved to use DNA for long-term storage and proteins for heavy lifting. RNA is the ancestor. But Cech doesn't just leave us in the primordial soup, he brings it back to the present. Because if RNA is this powerful, if it can cut, splice, and code, we can use it. We saw this with the mRNA vaccines for COVID. That was just a piece of code instructing our cells to build a spike protein. But there's also CRISPR. We usually talk about the Cas9 protein that cuts DNA. But how does Cas9 know times where times to cut? It uses a guide RNA. Again, the protein is the muscle, but RNA is the brain. And then there are telomeres, the caps on our chromosomes that keep us from aging. They are rebuilt by telomerase, which, you guessed it, carries its own RNA template. It's everywhere.
It really is everywhere. And Cech makes a compelling case that we are just getting started. He mentions that there are thousands of non-coding RNAs in our cells that we used to call junk. We thought they were trash. Now we know they might be regulating our genes, fighting diseases, or even causing them. We are moving from the age of DNA, where we just read the static blueprint, to the age of RNA, where we can actively edit, manage, and rewrite the processes of life. It's dynamic. It's fluid. It's the difference between having a library of books, DNA, and having an active internet where information is constantly moving and changing, RNA.
So, the backup singer is actually the genius composer, the lead guitarist, and the tour manager all rolled into one. I think I'm sold. Sarah? It makes you look at biology differently. We aren't just hard-coded machines. We are these fluid, RNA-driven entities. The catalyst is a perfect title because RNA catalyzed life itself, and now it's catalyzing a revolution in medicine. It's been a fascinating ride from the Wool Tie Club to the Nobel Prize. Thanks for taking me through it.
Anytime, Alex. And to our listeners, if you want to understand the technology that is going to define the next 50 years of healthcare and maybe even explain how we all got here in the first place, pick up Thomas Cech's The Catalyst. It's a deep dive, but hey, that's what we love. Thanks for listening to our show. Keep asking questions, keep being curious, and remember, sometimes the most important things are the ones hiding in plain sight. Or, in this case, hiding in the junk of our cells. See you next time.
Outro music fades in. Absolutely. And just a quick final thought on that junk DNA idea. It really teaches us humility, doesn't it? We labeled 98% of our genome as junk just because we didn't understand it yet. It turns out that junk was the operating system. It's a good reminder for science and life in general. Don't dismiss what you don't understand. All right, that's a wrap. Catch you on the next episode.